Welcome to today's webinar. This is Nick Sorbo of the Science Collaborative. On today's session, we're taking a look at an interdisciplinary collaborative research project that examined eelgrass health and how that effort has increased trust among partners and inspired ideas for new management strategies by municipalities and state agencies. We have some folks from the project team joining us today to share more about that work, but as always, before we get started, we have a quick introduction to the reserve system, the Science Collaborative, and this series. The National Estuarine Research Reserve System is a national network of 30 reserves shown on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities you can see on the slide there, which include land stewardship, research and scientific monitoring, training programs for the public and local officials, and education. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. And the Collaborative Science for Estuaries series features project teams, as we have today, supported by the Science Collaborative program staff, and partners. And with that, I would like to introduce our speakers for today. Bill McDowell is an ecosystem scientist and recently retired professor in UNH's Department of Natural Resources and the Environment. Lynn Vaccaro is the Training and Engagement Coordinator at the Great Bay National Estuarine Research Reserve in New Hampshire, and Kala Matzo is the Director of the Piscataqua Region Estuaries Partnership. And as we transition to our first speaker, we have a quick open-ended question for folks in the audience, and you can just answer in chat. What aspect of today's webinar interests you?
Yeah. Thanks, Nick. It's fun to be on a webinar with you again. Yeah, and we are sincerely interested if anybody wants to type in some responses to this chat. It's just fun to know kind of who we're talking to and what interest you have. Restoration, collaborative process with communities, eelgrass and collaborative process, all of it. Thanks, Megan. Water quality, fun. All right, well, it's motivating and makes it feel more personal to see a little bit of that, those comments in chat, so thank you. We got some Great Bay people, yay. So this is our outline for today. I'm going to start by sharing a little bit about the management context for this project and the collaborative process. Then Bill's going to walk you through the research approach and the findings. And then Kala's going to wrap up with some reflections on the lessons learned, benefits, and next steps. And hopefully, if we're on time, we'll have about 15 minutes or so for questions at the end. And it seems like, based on the comments I see, there's going to be something for everybody in this presentation. I'm glad to see there's at least a few people interested in the collaborative process as well, because that's what I'm going to talk about. So the Great Bay Eelgrass Resilience Project was, as Nick said, was a 3-year collaborative research project funded by the Science Collaborative. We wrapped up in this past fall. And we really wanted to improve understanding of eelgrass resilience in Great Bay in New Hampshire, and also fill some gaps that we thought could help, better manage nitrogen levels in Great Bay. And probably kind of an equally important goal that was maybe a little less explicit was that we hope to also, build consensus and trust around this issue that had been somewhat controversial in Great Bay. So these are all of the folks that were involved in some way in the project. You can see we had a really big project team, including Corey Riley, who is the co-collaborative lead and is on this webinar too. And Anna Mikulis, who is a graduate student and a project manager for the project, and hopefully you'll hear her voice a little bit in the Q&A. And then we had a really terrific advisory group and some external advisors who supported us. And so we really took to heart these principles of collaborative science, that are really nicely explained in the Guide to Collaborative Science. And in fact, we used these principles to guide some of our early meetings with both the project team and then again with the project advisory group, just to make sure everybody was on the same page, that this is something we had to do. This were expectations of the funder, and we were going to do our best to kind of, embody and embrace these principles. So the management context was very front and center for us, and is, probably a somewhat familiar story for those of you who work in other estuaries around the country. In 2009, the Great Bay Estuary was listed as nitrogen impaired. In 2021, after a lot of research and debate, EPA established kind of a regional permitting structure called a Total Nitrogen General Permit, with the expectation that that permit was going to be renewed in 2026 with the possibility that it could be made a little bit more stringent. And so one of our early advisory group meetings, we invited Ted Deers, who was on our advisory group and is a senior administrator with the Department of Environmental Services, and so was deeply invested in water quality management in Great Bay, to share with the group tell us a little bit about, the management context from your perspective, and where do you see this project fitting in? And so this is kind of how he responded. He put together a few slides for us, and I've just got two of them here to show you today. To TMDL or not to TMDL? That is the question, which also gives you a little taste of his sense of humor. And TMDL stands for total maximum daily load, and so would have represents kind of a more stringent, more rigorous way of managing pollutants in water bodies. And it's not an approach that has been taken in Great Bay yet, but it was always sort of looming. And so the hope was that this project could, kind of weigh in and help kind of guide some decision making. Ted further explained to the group in good New Hampshire dialect that it's wicked complicated, that there's lots and lots of variables to consider when you're thinking about eelgrass and nitrogen in Great Bay. And so we hoped that our project could help kind of illuminate some of the connections between these variables, and maybe provide some of the numbers or the parameters that could be useful if you had to build a very quantitative water quality model to generate a TMDL, in the case that they would go that direction. So given all of that, we knew that we really needed to be kind of careful with relationships and who we engaged in this project. We built a pretty big project advisory committee that included state and federal water quality regulators, the municipalities who were regulated by the permit, folks who thought they could use the science for restoration, and it sounds like some folks on the webinar today also have that interest, and then others who are just champions and advocates for Great Bay. And I'll note that Great Bay really, we were part of the project team, but we're also end users, and so we kind of fit nicely in these last kind of two categories, I would say. And we also had a big project team that had kind of some sub teams within it. We had an executive committee that kind of helped integrate the technical and collaborative aspects that provided a really nice kind of way to make decisions in a streamlined way. And then we did have some external advisors. And then we brought all those people together in what we called integration meetings, periodically throughout the project. So here's just a diagram of those integration meetings here in gray. We had about 6 in-person half-day meetings over the course of the project when we brought all those people together and really kind of hashed out what was going on in the science. Those are, of course, scheduled at times when we thought they could be really useful for providing input into decision-making, for example, planning for the next field season. And they also see in green here that we had some really neat kind of education work that kind of progressed in parallel with the research process and really relied heavily on the project team's expertise and maybe a little less on the advisory group's expertise. So here's just some pictures of what those meetings look like. Some were pretty traditional, but we also did get folks out into the field, which was really fun to just, this is like the deputy director of public works in Dover, who didn't often get a chance to go out on a boat and actually experience an eelgrass meadow, which of course is underwater for the majority of the time. And then we always started our meetings with this, just a quick recap of what we heard during the last meeting, which could have been 6, 8, 9 months ago, and then what we did with that feedback that we heard at the last meeting, just to really emphasize, that we were listening and using their feedback. And I'll just note this is a kind of an approach that I used when I worked with Jen Reed, actually, in my Michigan days. And then we were really careful to document everything on Google Drive, so it was very accessible from meeting to meeting. And then we also heard from our advisory group that they felt very compelled to keep their community, their organization, abreast of the project. So we did do a little newsletter, kind of like I've seen other research projects do, like the eDNA project did the same approach, that provided just a nice quick summary of the status of the project that they could forward on to the other people in their organization that also wanted to kind of follow along. And then we had some, really management relevant products in mind, some maps of water flow parameters, some really specific analyses we promised to produce. And then we also promised to, go back to those original policy questions and distill the science and be really explicit about how well did the science address these specific management questions. And so we called that our so what product at the end. And then I'll just end with kind of this picture here of one of the products that came out of the education part of this project. Our education director, Kelly, was able to kind of leverage the science and get some additional funding to do a whole update of our visitor center that included an eelgrass exhibit, which was really fun. So I think with that, I'm going to pass it over to Bill to dig into the technical details.
Great. Thanks very much, Lynn. And welcome, everyone. It's a pleasure to present this information to you. And I hope you find it interesting and exciting as we have found it to be very exciting as a project to work on. And so in the big picture, the decline in eelgrass in the Great Bay Estuary has been one of the key drivers of our interest in studying the bay as a whole ecosystem. So this is a rather sobering graph that from 1996 to 2022 shows we've lost 65% of eelgrass cover over those 27 years. And, you know, some years perhaps weather has played a role in a good year or a bad year, but the long-term trend is unmistakable and unfortunate. And so this was really the motivation, one of the key motivations behind our assembling a project team and building the proposal that we then submitted and ultimately got funded. So I think it's important with the broad audience, especially that we have today, it's important to set the context for understanding hypothesis-driven research. Fundamentally, rejecting the hypothesis is how we make progress, you know? And so if we fail to reject the hypothesis, well, we can make new predictions and figure out what we think is going on in the system. And so we start with observations in this process. And the observation I've already shown you, eelgrass is declining. And then from that, we might make some questions. Why is an obvious one. And then we might propose some research activities, understanding the algal composition in the bay, which is known from other places to affect eelgrass health and viability, understand the water quality in the bay, which can have negative effects on eelgrass. And so from these research questions, we can generate some specific hypotheses that we test. And then we get into even more detailed aspects of understanding the system with various sorts of subtests and specific sub-hypotheses. Putting this all together, we're really trying to understand what the drivers are of the system as a whole. What leads to good years for eelgrass, what leads to bad years for eelgrass, and how do in a given year, or a few years in the case of this proposal, how do these components that might be affecting eelgrass, how are they interacting on the ground in the field? And what can we then learn from those results about how the system works? And so in our project, we posed two research questions. Research question one was, does eelgrass health vary spatially in response to variability in water residence time, bed shear stress, how fast the water is moving back and forth across the eelgrass bed, that load of algae or epiphytes in the system, and the water quality. And we can frame this in the simplest terms as understanding how the physical environment affects eelgrass. And research question two kind of flips that around, where we examine how eelgrass affects the physical environment. And so we can think of this as some of the self-healing aspects of eelgrass, in that the more eelgrass we have, typically the better the water quality, the better the sediment deposition in the eelgrass bed, and the better the eelgrass does. So the better it does, the better it continues to do. The flip side, of course, is when it's on a decline, it has the possibility of degrading over time as it's able to heal less and less over time. So some of the key aspects of the eelgrass bed function are nitrogen retention and loss, removal of nitrogen from the system to the atmosphere, and the deposition of sediment or sediment filtration. And so we wanted to look hard at how some of those properties vary along hydrodynamic flow paths that span from unvegetated areas to vegetated. So we wanted to see if there were differences in the eelgrass rich areas and the mud flats that had little to no eelgrass in the system. And so our research approach in our original reviews, it was like, oh, this is really ambitious, to the point of like, no, you can't really do this. We're testing multiple hypotheses about multiple stressors, and we're going to provide a new perspective on hydrodynamic behavior of the estuary. And we're going to improve understanding of nitrogen cycling rates and denitrification. And in fact, it was a very ambitious project. We largely succeeded in pulling it off. And the reason was that we had extensive data from that set the stage. We had extensive ongoing sampling that we could piggyback off of. And the fact that we had an excellent call out my excellent, now PhD, Dr. Anna Mikulis, who did most of the work that we're seeing, the field work that we see in this project, as well as worked very closely with me and the team in devising the project. And so ambitious and also embedded in the community context. And so here are some of our research questions. I'm not going to go through each of the individual hypotheses, but research question 1, does eelgrass health vary spatially in response to variability in water residence time, bed shear stress, algae, epiphytes, and on, water quality? And so we'll just highlight the very first specific hypothesis. Eelgrass health is better at sites, these are what we call tier 2 sites, that have longer within meadow residence time. So we're combining our hydrodynamic modeling with our understanding of how the system is working. And so this leveraging of existing programs occurred across 3 really pretty distinct environments. The Portsmouth Harbor area, so off to the right at the bottom, is the Atlantic Ocean, you know, that's the mouth out to the ocean. The Little Bay Piscataqua River is up in the middle there in the north, and Great Bay in the left-hand corner of your slide. And Great Bay is where we focused a lot of our effort, because it's has the potential to hold a lot of eelgrass and provide a lot of ecosystem services, but it is stressed right now. And so this tiered seagrass monitoring approach is based on the work of Nichols et al. 2012 paper. And basically, we did, all tier 1 is a general assessment of the bay. Tier 2 is an ongoing program that Dr. Mazzo is continuing to lead, that looks at percent cover of eelgrass, canopy height. And then tier 3 is where we got much more detailed assessment in individual beds. And we used that then to test some of our specific hypotheses about how well the eelgrass is doing and what is a related and potentially driving a good or a bad outcome in terms of maintaining the bed. And so here we're showing you just some of the samples that we took and showing our research outputs by the numbers. So if we start on the filters on the right, so this represents a little aluminum foil cup. And in the cup, the white circle is a glass fiber filter. So we filtered water from the bay, from different parts of those 3 regions of the whole Great Bay Estuary system. And you can see that there's, for example, number 36, site 36 in the middle and the upper row has very little dark matter on that filter. And 68 just below it has a lot. And so this suggests that well, we're documenting here that there is a difference in the total suspended solids after we weigh them and quantify that. So this is the kind of variability we see across our system. And so we established 25 new eelgrass monitoring sites. 300 quadrats were assessed during the summer sampling of 21, 22, 23. 70 water quality samples were collected and analyzed specifically for this project beyond what other samples that are already analyzed for the bay in general. 48 samples for total suspended solids and 30 light casts to measure the light extinction, or KD. So we have then the 70 paired water chemistry and eelgrass seaweed measurements across 3 summers. And we're going to use these data to test our research, to provide various analyses to answer our question of research question 1, how does physical environment affect the eelgrass? And we can see here that in this correlation matrix that there are a lot of variables measured, and the point we want to look at is, let's take eelgrass density. Maybe you can see my cursor. Eelgrass density against eelgrass epiphyte load is actually a strong correlation. And eelgrass biomass is closely tied to the eelgrass canopy height. Well, that makes sense. The epiphyte load response makes sense too, because we can't have epiphytes on eelgrass unless there's a lot of eelgrass in the quadrat. So I'd like to highlight 2 findings here, though. If we look at this plot, this result rather, for eelgrass biomass, and how well is that correlated with the dissolved inorganic nitrogen in the water, and how well is the seaweed, is eelgrass density correlated, the upper part of this red circle, with the inorganic nitrogen. And we see that it's a nitrogen impaired estuary, we're told, and we do believe. And yet at the same time, when we go out and measure the inorganic nitrogen in the system, it's not a good predictor of how strong and how robust the eelgrass is at a given point. And this is because we might think of that nitrogen as just what's left over and used. The most available inorganic nitrogen has been used up. And if we look to the right of that red circle, we see some statistically significant relationships between the eelgrass biomass and the total nitrogen and the dissolved or organic nitrogen. And so the total amount of nitrogen in the system is related to the eelgrass metrics, but not the inorganic nitrogen. So that's an important point, because it's only recently that we've started to parse out, well, particulate nitrogen is an important part, the organic nitrogen, the colored is a portion is an important part, as well as the inorganic. So I'd like to turn next to our hydrodynamic model of the Great Bay Estuary. And so this is one of the things we had promised that is really helping to set us apart in our understanding of the bay. And with the collaboration of our project team member, Dr. Tom Lippman, he both updated a model and provided new data for the model, but the use of drifters that were deployed in the first summer to try to fine-tune and better verify the model. And so the old perspective was that this is a well-flushed system, because the water really rushes in and out in the main channels, and it's definitely moving fast. But the new perspective from this work is that it's more like a lagoonal system. And we're going to show you some of the outputs that lead us to that conclusion. So with a lagoonal system, where there are parts of the bay, water just sort of relatively just sort of sits there compared to the well-flushed channel. And so we have some video here that follows particles that are put in one spot in the bay, the initial red, and now in 2 to 4 tidal cycles, this is where these particles end up. And the particles should be seen as, it could be a sediment particle, could be a parcel of water, it could be a nitrate ion. And what we see is that after that 50 hours, it's spread out all over if they're put in the channel down there in the southwest corner of the bay. So now let's look at the hydrodynamics from the southeastern corner of the bay. And they're just sort of sloshing around back and forth. And it's very different behavior from what we saw in the previous location. So what we see then is we can characterize different physical environments in the Great Bay based on these hydrodynamic characteristics. Now, another way to look at this is to examine how that is what happens to water which we place in the bay, in a bed within the bay, and how fast does it move out through the whole system. Another thing that we did with this hydrodynamic model is take individual beds, which we used as experimental sites, and say, well, how long has the water been within a meadow when we go sample it during the course of our experimental work? And we can see that in the eastern Great Bay, the place where the water tends to slosh back and forth, it could be 2 to 2.5 hours of contact within a meadow before it leaves, and the western is lower, and the northern up there, number 14, is even lower still. So we have a wide range of hydrodynamic conditions in these different sites. And so then this is an important finding and somewhat unexpected. Now, we leverage this understanding of the hydrodynamics to choose places with different flushing times and to go look at the nitrogen cycle, comparing the bare and the eelgrass rich portions of the bay, and understand all these or many of these processes. We didn't measure end fixation, for example, but we measured a lot of the pathways in the nitrogen cycle with the denitrification, the upward arrow that takes nitrogen out, and as N2 and N2O gas. We measured the uptake by seaweeds by looking at the growth of seaweeds, measured the uptake into eelgrass by looking at the growth of eelgrass over time. And this provides a lot of important fundamental information about how the system works. And then in these, given this background information, we decided to use the in-situ denitrification rates using push-pull incubations. Here's Dr. Mikulas on her paddleboard to try to understand the magnitude of the denitrification in the system. And what we see, these are data from the membrane inlet mass spectrometer, which we all in the business refer to as MIMS. Much easier to say MIMS than that. And basically, we add a tracer nitrogen and we look at its fate across the eelgrass and in the bare, as well as the eelgrass dominated beds, and measure how much denitrification is occurring and how much the potential is, how large the potential is for denitrification to occur on the right. And what we see here is surprisingly that the bare sediments and the seagrass sediments are no different. If anything, the bare might be a little bit higher. And this suggests then that the eelgrass is not having a dominant impact on its environment, and it is not playing a dominant role in driving denitrification as it is in some other systems. Well, to convince you of the some other systems is we've done this comparison with the from data by Aoki and McGlathery in South Bay along the Chesapeake, and what we see is that the Great Bay system, with its very high load of 900 micromoles N per meter squared compared to the 10 micromoles N per meter squared in the South Bay, the Bear sediment and the Eelgrass sediment have considerably higher denitrification than this more oligotrophic system. And so what's going on here? We think that denitrification rates, this loss of nitrogen, so it's a good thing. We're venting nitrogen out to the atmosphere, converting it to gases. It's leaving the system. Why are they higher in this eutrophic system than this relatively pristine seagrass meadow? We think that is because compared to South Bay, our Great Bay sites have 1.5 times the organic matter, which is an important part of generating conditions for denitrification, and 4 times more nitrate in the sediment. And so this provides a ready source of the conditions, the oxygenated waters to generate the nitrate, and pockets of de-oxygenated water or microsites where oxygenate is low, so that we can see the denitrification occurring. And so how did different drivers affect the eelgrass? We come back to this raft of specific questions that we had. And at the very beginning, that we didn't read through them all, but the hydrodynamics both seems to, it sort of cuts both directions. In terms of estuary wide, we see higher eelgrass biomass with a shorter residence times. So the short, fast moving water out of the coast, which is also clearer and colder and lower in colored material, that has the higher biomass of eelgrass with it, whereas within Great Bay, we see the opposite. Within the bay itself, higher eelgrass biomass occurs with the longer residence times, not the shorter residence times. We see algal competition, that the seaweed cover negatively correlates with eelgrass cover in the estuary-wide data set, but is not clearly related, as we saw in the correlation metrics with within the Great Bay itself. Estuary-wide, we see total nitrogen and dissolved organic nitrogen are strongly and negatively correlated with eelgrass biomass. So dark and nitrogen-rich water gives us lower eelgrass biomass, and that also is related to the light blocking availability. So summarize these scientific findings, we see that spatial variability in eelgrass health is only weakly related to the water quality above the eelgrass bed. This is our observational work and some experimental work on the rates of grass and algal growth. Second, Great Bay should be considered lagoonal and not well flushed. Third, the denitrification rates in both bare and vegetated sediments are very high relative to more oligotrophic estuaries, even though the eelgrass is not doing all that well. These results suggest that past nitrogen loading, which is probably driving our high sediment N content, along with the high denitrification which we have observed, it may mean that we will see delayed but ongoing response to recent reductions in nitrogen inputs from the wastewater treatment plants. So the denitrification is a big, it's an important process, but it is not yet sufficient to greatly reduce the nitrogen concentration in the water, nor is it sufficient to foster eelgrass health at this point. And next, I'm going to turn it over to Dr. Mazzo.
Thanks so much, Bill. Hi, everybody. So great to be here talking about collaborative science. This picture was just taken in the Great Bay actually just a few days ago, so I had to include it. And if you've seen something like this happen, you know that the crabs are moving all over each other, the water's muddy, there's a lot of conflict, competition for females. egg-laying sites. And between 2010 and 2020, we were experiencing sort of a human version of this around the subject of nitrogen. And in fact, I would say it was more, dare I say, violent than what you would see here with the horseshoe crabs. There was a lot of verbal attacks between different people being involved. And in one case, I had a consultant come up from Stony Brook University, and I warned him, things can get pretty heated around here. And he said, Kala, I've been doing this for decades. Really, don't. I know how to handle myself. He came up to me after the meeting. His eyes were as wide as saucers. And he said, I have never experienced what I just experienced from such and such a consultant. So you have to understand if a lot of this, what you might sit here and think as a scientist, wow, there's so much collaboration involved. You got Lynn and Corey and Kala, and so much being poured into the collaboration work. It had to be this way because of the amount of distrust that was in this system. So we tried something really different, and I'm curious if people could put in the chat if they've ever experienced or heard of anything like this. We decided to have a real-time peer review as part of our project, and our peer reviewers were not from around here. So you can see there where our peer reviewers were from, a couple from Canada. Unfortunately, Brian Howes died in the first year of our project, but he helped us actually write the proposal. But so it's important to understand that the peer reviewers were talked to before every one of those meetings that Lynn mentioned earlier. And then they were at the actual meetings so they could respond to what they were hearing, questions from the lay folk and statements by the local scientists. And then we would talk to them afterwards. And there was a tremendous amount of work around the involvement of the external peer reviewers. There was some concern that they might actually, the whole purpose of having them is to increase the credibility of the work. Credibility being defined as the perception that the technical work is adequate. It meets standards of technical adequacy. And so I was really worried that if there was too much back and forth, it would get confusing to the non-scientists in the group. But what we saw was when we got a lot of people together to talk about science, to watch science, you see Corey there and Dr. McCoulis and a consultant there in the white, I don't know, is that a cardigan? And some municipal folks. So what we found, and was very surprising to me, was in talking to people after meetings that were particularly back and forth between the scientists, in some cases disagreeing with each other, I was extremely concerned that maybe we were going to see a decrease in credibility, the perception of credibility. And it was a huge surprise to me. And I've done a lot of work in interviewing with people on this topic in the past. And everybody to a person said, Kala, it was so good to see how you disagree and see how professionally you disagree. And I feel like I trust the science more now. And I was blown away by that result. Next slide, please. So this is the other reason that you have external reviewers is because we get locked into our perspectives and we get locked into our biases. And it's once you get locked into a perspective, it's very difficult to shake it. Some of you might look at this picture and see a young woman. Some of you might look at this picture and see an old woman. Once you've your brain has made a choice, it's very hard to switch. But in fact, you can see both. Someone who's incredibly broad-minded, like Corey Riley, instantly sees both the young woman and the old woman. But it can be really difficult. And we heard comments from our external advisors that I've been part of these conversations for 15, 20 years, I had never heard people say before. And that's what happens when you bring people in from outside of your neighborhood. As an example, so much of our conversation is always focused on what made the eelgrass go away. We had one of our external advisors stand up and say, you know, everyone's focused on what made the eelgrass go away. I'm less interested in that. What I'm noticing is that you have lost 80% of your oysters and 60% of your eelgrass since the early 90s. The takeaway message here is your system has lost the ability to process the loads that are going in. And I looked around the room and you could just see everybody just had a little paradigm shift in their brain. But nobody had said that before. So I just want to kind of close by thinking about a metaphor. If we liken a collaborative science project to a play, my son is a playwright, so this was in my brain. What would you consider that it's the costume? Is it the script? Is it the actors? After this incredible experience working with everybody that you see on your screen right now, I would throw out there that collaborative science is the stage. It becomes the foundation for people to explore really difficult concepts. And so, you know, Corey and I were talking about this the other day. We had so many, as Bill said, it was extremely ambitious. And 2 of our 3 years were some of the wettest years on record, which was very challenging for Dr. McCoulis and her field work. But what I found from this project is if you invest enough in the collaboration, then you can still get a tremendous amount in terms of moving the management needle. And I have seen this in my work. When I sit with the municipalities, since this project has ended, I see an increase in trust. And so to me, the investment in building that strong foundation is absolutely paramount. So just kind of going over all the slides, you know, the collaborative process is intense. We had a lot of really hard conversations, but they were worthwhile. And, you know, we got some great comments about how accessible the science was to folks. And people were happy to see the exchange of ideas and even to see us disagree. But again, that was because it was done in a very planful and collaborative way. We had so much leverage support from all the different entities that were involved. And if we were to do this again, we would probably streamline and separate the groups a little bit and incorporate more formative evaluation. Research insights that you heard from Bill, completely new perspective on hydrodynamics, and it's very, very paradigm changing. Because until this project, people would just say, well, it doesn't matter. The phytoplankton, it doesn't really matter that there's too much nitrogen causing phytoplankton blooms, because it all just get washed out to the Atlantic in 2 days. And it was very hard to dispel that bias. But you show people that video and it's dispelled. And as Bill said, the denitrification rates are quite high. And it shows the promise of what a healthy eelgrass bed can do. But we certainly don't have an healthy eelgrass bed right now. And so management insights, this idea, and again, if you've seen other projects try this, maybe Nick, who's part of the Near Science Collaborative, have you seen simultaneous external peer review in a project before? We hadn't seen it. It was really challenging, but we believe it was worth it.
Great work, and thank you, everybody. I think we're going to transition to Q&A now, and I'm going to put us in gallery view. And folks who are in the audience, you might see there's a few extra faces here. That's because we have more people to help answer questions as a little treat. So bonus panelists, you know, love to see it. We are monitoring the Q&A and the chat for questions. So put questions in there if you have them. Don't be shy. We've got quite a bit of time to answer questions. We had a few come in already. One of them was a point of clarification. Somebody had asked what the tidal range of Great Bay was. I think I'll answer 2 to 4 meters. So just for that clarification. So to get started here, we have a question. Does the uptake of nitrogen by phytoplankton and macroalgae provide another route for nitrogen to sediments via microbial decay, which would foil the correlation between water concentration data?
Yes, absolutely. To answer that question, that pathway definitely exists. We did not look at phytoplankton directly during this project, but we did look at uptake of nitrogen by 2 of the common species of seaweed in our system. And as a relative percentage to overall nitrogen input to the system, uptake by seaweed was very little, like less than 1%. And so while it represents a pathway, I don't view it to be a significant pathway necessarily.
If I could follow Nick, I would just say the other thing in context is that, as I mentioned before, 2 out of these 3 years were very, very wet. And so primary producers were squashed across the board. In 2023, I don't think we ever saw higher than maybe 7 or 8 micrograms per liter of phytoplankton. There was very, very little seaweed biomass. In 2021, the 1 year where we had pretty good light, there were several algal blooms above 20 micrograms per liter.
Of chlorophyll A.
Chlorophyll A, thank you.
Okay. Anybody have anything else to add? No. Okay. We had another question earlier in the presentation. Somebody had asked what the dispersion model was that was used. Colin noted in the chat that the model is based on the regional ocean modeling system. I just wondered if you had anything else to add, either you or Bill.
If anybody had a specific question, I think Tom Lippman might be on the call, and he would be the person to answer if somebody had a further question beyond just, the basic system that they used.
Looks like Tom is here. So I would also note too that if you have questions that you would prefer to verbalize instead of typing out, you can raise your hand and I can unmute you and you can answer your question verbally. So we can do that as well. And Tom confirmed ROM's model offline particle tracking in the chat.
Just to clarify, I think it's really important, and Tom can speak to this if people want him to, but I mean, so he actually put objects out in the water. Drifters. Couldn't remember the word. Thanks, Bill. So it's a model, but based on empirical data.
Okay, Tom, I just unmuted you if you want to speak more to the model.
Can you hear me?
We can hear you.
I don't know how to turn my camera on, but yeah, thanks everybody. Yeah, I'm happy to talk about the model. It's a ROMS model, the Regional Ocean Modeling System, with variable spatial resolution for the different models that we've run. Typically down to about 30 meters is pretty typical, but we also run it at 10-meter spatial resolution with vertical varying flow characteristics. We take that model output, save it at grid points every so often, and then we run offline particle tracking by imparting particles into a certain position in the estuary, and then we do a fairly sophisticated interpolation routine to then move those particles with the flow around it. And we add diffusion to the model by adding diffusivity coefficients to account for sub-grid scale motions. So the model tested really well for advection. We needed to estimate what those diffusivity coefficients were, and we used surface drifters in order to estimate that. Then we feel pretty confident that we can say something about how the model behavior affects how particles are transported through the estuaries. And that's what Bill was showing earlier in some of his simulations. And I could go on, but hopefully that answers the question.
Sounds like it did. Okay. We're still keeping an eye on both Q&A and chat, so, don't be shy if you have additional questions. I know the presenters were very thorough. I learned a lot. It's always fun to me to do these webinars because I only interact with a third of the teams, or I don't know at this point, maybe half, but there's the whole other slot of teams I don't talk to until it's time for a webinar, and then I get to learn a lot in a very concentrated dose, so that's always fun for me. But anyway. Okay, another question just came in. Do you believe that these models could also apply or be applied to trace element flushing within the Great Bay system? I'm not sure specifically who to pitch that to.
I'm guessing Tom.
Can people still hear me?
I can still hear you.
Yep.
Yeah, so so far as the trace elements could be model as a single particle, then it would be very similar to what we've shown before with other particles passively being transported by the flow. Those trace metals may also, as part of their life history, change in time and change chemically. That is a more difficult thing than to try to model, especially mercury and some of those other elements that have different species within the water column. But we feel like at least if they're passively drifting, that's certainly something that we could take a look at. Sedimentation within the estuary, a lot of times trace elements attach themselves to other particles. Sometimes those particles settle out in the estuary. That is a complex process when you have a mix of sand and mud, where hydrostatic forces tend to be critical in understanding how those particles fluctuate and then settle at the seafloor. Moreover, the presence of vegetation also impacts shear stresses, bed stresses, and how sediment is filtered in the water column. So it's a really difficult problem when you have chemically varying things or particles that settle out in the water column. There are lots of people who are pursuing that. We spent some significant time doing ecosystem modeling in other offshore systems, and we have experts. Jiangwen Choi, who helped develop these models, is an expert in that field. But we're not necessarily in our lab working on the trace metal problem at the moment.
Okay.
Nick, I can speak to Paul Stacy's comment if nobody else has a question.
Sure, go for it.
Yeah, Paul, in terms of the depth and the KD, I think you're onto something there. The other interesting thing is that the healthiest eelgrass is in Portsmouth Harbor, which is oftentimes growing in 8 meters of water, because KD is usually around 0.5. Whereas in the bay, KD, I would say, and Anna could correct me if I'm wrong, but very often it's between 2 and 4. So that turbidity of that really, really high organic content and extremely silty sediment versus the coarse sediment that you have out in Portsmouth Harbor, that turbidity is such a problem. In 2023, when we had such bad water quality, the birds, what kind of birds you got in that nest over there, Corey Riley?
Goldfinch.
Osprey.
Osprey, right? Not sparrows. I don't know why I was going to say that, but no, she has these beautiful, like an osprey cam, and for the first time, as far as I can remember, the osprey essentially stopped hunting in Great Bay because they couldn't see anything. The water turned to creme brulee, and that is not a nitrogen problem solely. In fact, mostly it's a TSS and a CDOM problem. So, but as Bill mentioned earlier, out in Portsmouth Harbor, you have a very different system.
We've got a few minutes left, so we have time for like maybe one more question. If no one else has one, we definitely have one that we always want to know about, that we rarely get to ask, which is what comes next from your perspective, just in either the science or collaboration fronts for this project. I see Paul also had another question that we can maybe answer afterwards too.
I could try something unless Bill and Anna, Corey, Lynn, do you guys want to go first? Okay. So what was really amazing about this project was we were able to do some things that had never been done before. A lot of Anna's work, getting into the sediment, looking at the pore water, measuring denitrification rates, process rates. This enables us to start to move towards a more comprehensive model of what's going on out there. But the other thing we want to do now is take a step back. Some of the relationships that we saw over the last 3 years, what happens when you look at those same relationships over 40 years? We have, thanks to Fred Short, we have an eelgrass data set going back to the early 80s. And we're trying to rescue as much of the older data as we can and do some statistical work to look at what's been happening over sort of the decadal scale versus just the 2 to 3 year scale. So I think that's one thing that's next. But really keeping up the strong collaboration that this project began. Having lived through what we call the nitrogen wars, I'm very motivated to keep things on the really good terms that they are on now.
So Carlo, it might be worth also reflecting on what's next. Is we are also started as sort of a spin-off, but it's a completely separate project with our municipalities, trying to better understand the source of the particles in the bay. You know, they may be coming in from the rivers, they may be generated by resuspension. And so we've done sampling last year, we're continuing this year to try to assess that, because the particle load is very significant in terms of, degrading the light environment for the eelgrass.
That's really interesting.
Thanks, Bill.
Somebody added some suggestions for what you can study next too. Some surfactants from boat soap should be studied next. So you got some advice from the audience too if you are looking for other things to look into. We have like 2 more minutes left, so if you want, we can try to tackle this last question from Paul. The most difficult management challenge is translating ambient concentration data into loading limits, and then figuring out how to reduce those loads, especially from the landscape sources. How's that going?
I could take a stab.
Yeah, go for it.
Again, coming back to this idea of the collaborative science being the stage, at our last meeting, we were really good, I think, about making time for people to just sit around and talk. We broke people up into breakouts. And one of them was around sort of management and targets and TMDLs. And I was blown away because as we started to zero in on how are we going to get to targets, some of the folks at the municipality said, I really hope you don't come up with targets, because the existing target right now, loading target, is very difficult for us to get to. And we're collaborating right now. If you put out a new number, we're going to start suing each other again. So can we just keep collaborating for a while before coming up with a target that we're all going to disagree about? And I said, okay, interesting. And I looked to the regulators, and they were basically like, we agree. Like, we need more information. This project was great, but we need to do more work. We're not in a rush to set up a TMDL or a target quite yet. Everybody's working really hard to bring down the nitrogen loading. We've reduced point sources by 80%, and now everyone's switching to start thinking about non-point sources. So it was this great point about collaborative processes, where all of a sudden we found that our whole group was like, actually, we don't need to rush into a TMDL right now.
That's fascinating.
Yeah, and unexpected.
Yeah, I would say so. We are
And
out of time, so thank you to everybody who joined and submitted questions and followed along with the presentation today. A special thank you to our presenters for doing such a great job prepping for and putting on this presentation. As far as next steps go, as soon as you leave the session, and also tomorrow at the same time, you'll get an automated email from us via Zoom with the exit survey and follow-up link to resources, basically, so you can see the summary page with the recording, which usually takes us about a day or two to process, no more than a couple days. So you can look out for the recording there. We have another webinar coming up on June 20th, and then after that, July 23rd. You can look out for announcements for those, and also check the Science Collaborative website for that. And with that, just again, big thank you to all the presenters. I hope everyone has a great rest of your day, and everyone hang in there and take care. And presenters, no need to stick around. I will follow up with you separately via email. Thank you again for your great work.
Thanks, Nick.
Thanks, everybody.